Alright guys, more forensic entomology notes. It's all about the bugs. So we got a cute little cartoon here. Hope you guys enjoy it. Alright, so something you probably already know. Insects are the most diverse and abundant forms of life on the earth. There are over 1 million described species. More than 2/3 of all the other known organisms out there, and they have impacts on human lives in lots of fun-filled and exciting ways. Probably more than what you really want to know. Forensic entomology is the use of insects and other arthropods that feed on decaying remains to aid legal investigations. There are all sorts of ways to do it. You can look at urban legal proceedings involving insects and related animals that affect man-made structures and other aspects of the human environment, such as bedbugs and termites. This could be criminal and civil law. There's medico-legal, which is criminal. And you have storage product pests, or civil, where you're looking at food contamination. So it's not just what you normally think about. It's actually a variety of situations here. So where can you see it? You can see it in homicides, abuse and neglect of human and children and the elderly, animals. Looking at transportation history, possibly. Cause and location, details of accidents, negligence, civil duty. So lots of different ways you're actually going to find this being used, a lot more than what you think about. The medical legal forensic entomology looks at violent crimes, which is what we've been kind of really doing heavily in here. They can establish timelines, your PMI, movement of the corpse, manner and cause of death, association of suspects with death scene, detection of toxins or drugs. You can do that by looking at the insect larvae. Since they're feeding on the corpse, they're also feeding on the drugs. So we already know about the time available. We kind of talked about that continually. What we do know also, we talked about, is the stages of decomposition, looking at the insect community, because some insects go faster than others, and some actually like different parts of the body and different parts of the stages of decomposition. Whoops, I don't know why I did that. Let's go back over here. All right, once a person dies, his or her body part starts to decompose. It starts with the action of microorganisms such as fungus and bacteria. Then you got your insects coming in. Your bodies are going to go fast or slow depending on weather conditions, which we talked about already. Then we got the stages. Now this particular one talks about the 4 stages, and it really depends upon where you're at, how many stages there are. But we do know at every stage you got different organisms being attracted. So we talked about algo mortis already, and liver mortis and rigor mortis. So I'm not going to beat that one to the death anymore, because we've gone over and over and over it. Here's a nice table for you talking about estimating the time of death. Within 4 minutes after death, you got decomposition hanging out, starting and ready. So your cells are already deprived of oxygen and energy. Carbon dioxide in the blood increases. pH decreases. Waste starts to accumulate. All these little guys are poisonous to the cells. Enzymes are dissolving, and you have autolysis starting. Putrefaction, destruction of the body proteins, your soft tissues by the anaerobic bacteria. First sign is that lovely greenish skin. Then you also got released gases like methane and ammonia that causes bloating of the body. And you also got that wonderful unpleasant smell. Here's a really nice chart that talks about all the different decaying. And here you've got little bit more than the 4 stages. A lot of people will put these 2 into separate stages here, but it gives you all the details of it. And you can stop the video and take a look at it. So when you got a fresh body, some of the ones that come in there immediately are your caliphorid flies, especially the green bottle flies. They're first to arrive and lay those fun-filled, exciting eggs. Your hairy maggot blowflies, which is also another caliphorid fly, come on in, usually the second to arrive. The bloating begins 4 to 10 days after death. At this point, blowfly eggs have hatched and the maggots are in the orifices. So remember your eyes, your nose, your ears, your mouth. So your histrid beetles now appear under the body, if the body's in land. Could be in the water. Butyric formation, your fermentation, about 10 to 25 days after death, and maggots are starting to leave the body and pupate in the soil. Now you got your carrion beetles and your skin beetles. They become the dominant necrophage, eater of the dead. Your dry decay or skeletonization is 25 to 50 days after death. They can last for years. You got dry skin, hair, and bones remaining. Now your fungus, your beetles, your mites, your clothes moths are most common. And some of the normal soil fauna, the bugs in the soils, the animals in the soil begin to inhabit the body. So you got some nice pictures here, 1, 2, 3, 4, 5, and 6. So you got your stages here. This is the pig. And if you're interested, you can try loading up some of these YouTube videos for yourself and see if they are actually running or not. You never know. Some of the factors affecting decomposition: temperature, humidity, which we talked about. Access to the body: is it in water, submerged? Is it on land? Is it in the shade, not in the shade? Large wounds lead to faster decomposition. Have scavengers and predators come on into the situation. Is it covered, tightly wrapped up, heavy clothing? Is the person high in fat or skinny minis? Insects on coke or meth burn through a body faster than those that are not. Strange but true. Bodies with arsenic decompose slower. So your decay rates are variable here. When the Body Farm did a study using 150 corpses, they took a look at, and the most important environmental factors in corpse decay were temperature, access by insects, and how deep something was buried. Other factors that were important were chemical, like embalming, insecticides, lime, that sort of stuff, and animals actually disrupting the corpse, getting in there and pulling their stuff apart. So we got some interesting cases here. I want you to take some time and read them. So you can stop the video anytime you want to. Take a look at some of these cases and what was going on. So we got some more terms here. Functional roles of insect associates of cadavers. You got your necrophages, blowflies, some beetles, your facultative necrophages, your ants, your cockroaches, some flies, some beetles. And then you got the enemies of the necrophages, your yellow jackets and your ants, and your inventives. These are the accidental species with no actual feeding relationship to the cadaver. They're just kind of there. So your colonization occurs in different stages. Different groups are adapted to different decompositional stages of the corpse, and they're fairly predictable, which is nice if you're trying to figure things out. So your major groups of necrophages, you got your flies, so blowflies, flesh flies, house flies, and something later on, your cheese skippers. And then your beetles early and mid through it. You got your carrion beetles, and then your dermestatids later on. And then I got your family of insects here and some examples of what goes on with them. Once again, more of these. You can stop there and read some of these. So now we got some really great pictures here of the fresh stage of the bodies where blowflies are starting to get into the cadaver and eggs are being laid. And you got your bloated stage here. Got your flesh flying, appearing, small maggots are feeding. Got your decay stage where the blowflies are just not, they're like, I'm done with this. This is not really all that tasty anymore. You got your maggots feeding. Your predators and other maggots arrive. And then you got your cadaver drying out. And the maggots are like, we're done with this. So blowflies are the most common insects associated with the dead body, but there are lots of other species. And you can use all of them to figure out their PMI. So now you got beetles here. Got some definitions for you: necrophages, omnivores, parasites, and predators, and incidentals. Got a really nice life cycle here. One of the things we talked about, temperature is especially important here. Most range for entomologists between just 2 and 4 weeks where they're really useful. Information and succession of insects in a corpse for you. And some lovely pictures of your blow and green bottle flies, your flesh fly, your cheese skipper, house fly, carrion beetles, road beetles, clown beetles, hide beetles, skin beetles, ham and checkered beetles. Boy, they got some fun and exciting names here. More information on blowflies for you. Feel free to stop the video and take a look at it. Once again, more detailed information for you. Please feel free to stop the video and look over this right here. Take some time to read it. So I've got some stuff here for about collections. So what you need to collect for forensic investigations. You've got to be very careful when you start your whole collection situation. You got to take a look at the surroundings. Look if it's outdoors. Look at the landscape, the plants, the soil types. Look at the weather, the temperature. You got to take a soil sample. And then if it's indoors, you got to look at the access points. How can the bug get in? Once at the body, the forensic entomologist takes several samples of different areas. You collect the maggots. You collect the eggs. You collect the pupal casings. And then at this point, you start to figure out your evidence. Once again, more information.